I want to start by introducing our first speaker, which is Simone Coppolino. Simone has a bachelor degree in mathematics and a master's degree in computer engineering. He is currently a researcher at the Institute of Biophysics of the National Research Council in Palermo, Italy. And in the talk today, he will present us his work regarding neural networks based on hippocampal circuitry for robot spatial navigation. Again, feel free to send questions even during the talk in the chat, and Simone, the stage is yours. Good luck.
Thank you, Ziv, for this presentation. I share my screen. So, my research project involved in the spiking neural network implementation is spiking neural network for robot navigation based on hippocampal circuitry. First of all, after talking about the model itself, I want to discuss about the biological background behind the model. The entire model, in fact, is inspired by hippocampal circuitry. The model implements some important biological concepts, such as place cells, and direction cells, and persistent firing neurons. Place cells are responsible in the creation of spatial maps in mammals. They are particular neurons that fires when mammal is in a specific location or see a particular object. A direction cells encode the angle of the head. A direction and place cells cooperate in the spatial representation of the environment. Persistent firing neurons are responsible in the creation of working memory. They are existent and confirmed in the hippocampus. Of course, not all the hippocampus is implemented, but only a subset of its circuit. In fact, in the image on the right, you will see the white section is the section that I implement in the model of hippocampus. On the left, you will see the learning environment. It's a simple path, but the most important things are these virtual, colored monitors. They are markers. The network is able to recognize 4 known colors and learn them. In fact, the network can learn the existence and the position of each marker to create a spatial representation of the environment. In a better word, the network is able to learn the markers to create an exit path from this simple environment. On the right, you will see the network. Is a network made by 21 leaky integrated firing neurons with the same electrophysiological properties. It's a feed-forward network with a balancing of static and dynamic synapses. The learning process of the network is through a symmetric spike time independent plasticity, STDP, rule. The first layer of these two bottom blue line is the sensor stream. The sensor stream takes the input from the robot camera, transforms it into a spike train, and sends it to persistent firing neurons and object neurons. The image from the robot camera is filtered because I want to find the number of pixels of the known color. In this case, the colors are red, blue, black, and green. The transfer function of the sensor stream is Gaussian. The reason behind this choice is more clear in the next slide. Object neurons are 4, one for each decoded color in the network. Their work is to fix the vision of a particular colored marker. In a better word, object neuron starts to fire when the robot sees for a long time, for a consistent time, a marker. Object neurons are connected with plastic synapses, STDP synapses, with all the place cells neurons. The place cells neurons made the output of the network. That is the input from the robot movement in the form of spike trains. Place cells takes the input from another input stream, the direction input stream. There are 12 place cells neuron. One of these place cells encodes a 30 degrees angle. So the robot receives the information about the object, with the existence of the object, and the angle of view of the object. So the robot has all the information to reach the marker. Persistent firing neuron, the bottom layer, implements the anticipation of the next visual input. There are 3 persistent neurons connected with all the other object neurons with STDP synapses. The persistent firing neurons takes the input from the sensor stream. In particular, when the sensor stream reaches the Gaussian peak, the persistent firing neuron starts to fire and continues to fire for 25 seconds. When the persistent firing neuron is activated, the connected object, for example the blue one, and its connection, for example display cells, are activated as well. This indicates to the robot the next direction to follow. So when the robot is seeing the red, for example, markers, the persistent firing neuron sends to the robot the next direction, in this case the object 1, to reach. The network has a section for obstacle avoidance, this part here, that interferes with the regular movement from the place cells. This is a simple description of the network. What are the major results and limitations of this model? The major results is the network is able to learn an arbitrarily long sequence in a single learning phase. This is the major results, also compared with other neural network, for example, recurrent or LSTM neural network that needs more than one learning phase. The second results connect to the first one is that the network is always able to detect the next visual input in the previous learned sequence. This is a very important result too. What are the limitations? The network is not able to learn correctly multiple paths in the learning phase. If the learning environment is a cross, the network is able to learn all the branches, but the network in the recall phase cannot choose what is the correct one for the X path. However, this limitation is almost resolved in an upgraded version of the model that are under developing in the neurorobotic platform too. The second limitation is that the network could reproduce, with some confusion, sequences with repeated object. If the learning environment has more than one same marker, the network can learn for a single object 2 or more place cells. The robot so receive the inputs from 2 or more different sources, and this can compromise the regular movement of the robot itself. So here there is a short video to show the functioning of the model itself. This is the learning phase. The robot made some bad movement, crash against the wall, but it doesn't matter. The most important thing is that the robot sees the markers and learned their position. So the robot made this movement until it reached the exit. The exit is made by the green one. In the recall phase, as you will see, the robot turns itself more earlier than the learning phase, because in this point, the persistent firing neuron starts to fire and indicates to the robot the next direction to follow. The same for the black one and so on. This is another recall phase where I put 2 obstacles to show the part of avoidance of obstacles, very simple. Also here. Further information and acknowledgement. The model involved in a paper on IEEE. The paper was approved at the end of December, and it was in press. In the model, in the paper, you will see the link to ModelDB to download the model and made some tests. The model is developed in the neurorobotic platform of Human Brain Project. The network is written in Pine and the transfer function in Python. I'm working in Biophysics Institute of National Research Council. My scientific responsible is Professor Michele Miglioret at Aix. And if you have any question about the model, don't hesitate to contact me at my institutional email, simone.coppolino@ibf.cnr.it. That's it. Thank you for the attention. If you have any question, don't hesitate.
Thank you very much, Simone, for the great talk. And I don't see any questions now, so I think we will move to the second talk. And after the second talk, we can discuss all the questions regarding both of the talks. I'd like to introduce you to our second speaker of today. Our second speaker is Alberto Vergani. Alberto has a master in cognitive science from the University of Milan, Italy, and has a PhD in computer science from the University of Insubria in Veracruz, Italy. He started his first postdoc at the Department of Computer Science at Middlesex University in London, and now he's doing his second postdoc in the Institute of Neuroscience in Marseille, France. And in his presentation today, Alberto will tell us about associative memory, modeling, and simulation. So, Alberto, good luck. The stage is yours.
Thank you, Dave, for the presentation. Can you hear me well? Right? No?
Yes.
Okay. So I start sharing. Okay. Do you see my PowerPoint? No. Okay. Yeah. Okay. So this is the first slide. So it's a sort of appreciation about this tea, coffee, and slides. So it's very cool. And so the topic of my talk is about associative memory. And the main example that I will propose is about hot coffee, not hot tea, but yeah. So at the end you see that it's pretty equivalent to see a hot coffee or a hot tea. Okay, so yeah, my email is this: is alberto.vergani@univ.amu.fr. And so this is a presentation about a paper that has been published last year. And the contribution of this paper is about my first postdoc position on Middlesex University in London. And the project was called NEAL, that means neuromorphic embodied agents that learn. And so the main topic of the project was about virtual agent. So build virtual agent in order to explore environment, to make association with memories, or make a sort of chat with the agent itself, and so on. And the model was about cell assembly. So is a neural network that acts as a virtual agent. And the specific topic of this talk is about associative memory and the bump attractor network. And so, as I said, you see the paper is this, and has been published last year. And at the end, I will show you also the link where you can download all the files and other information. Okay, so concept of representation in the brain. So how the brain represents concept is a very ancient question. And of course, there are also several questions important. And we focus specifically on how the brain represents physical concepts, like continuous value concepts, for example, pressure, force, temperature, or other ones. And also how the brain represents semantic, let's say categorical concept or discrete like, so unitary concept like what's a dog, a car, an house, a sun, and so on. And in this paper, we focus on a practical example. So because to do some advancement in research, you need to localize your research. And so we focus on a specific conceptualization. And we choose a very entry-level example. It is a beverage, judgments about a beverage, and some physical quantities. And in particular, we ask several questions, but the three ones are those. So how temperature grades are representing the brain, or how are represented and linked concepts like hot and coffee, or cold and coke, that are different discrete concepts. And how do the same hot concept manage the hot coffee meaning or the hot cock meaning? Because you know that hot is a very general attribution about an object, but it could be hot for one example or hot for another example. So it depends by some situation, but the same term is already the same. And so we adopt a model that is well known in neuroscience, or let's say in cognitive science, because there was a psychologist, Canadian psychologist called Donald Hebb, that proposed the cell assembly models, where it is a set of neurons that is able to represent a unitary concept. And we choose a different type of this cell assembly. The first one is the binary cell assembly. So basically you can have either an activation of an object or it stops, so it's deactivated. And then we have also bump cell assembly, where the concept is represented as a bump within the brain. So as let's say a fuzzy activation in the networks. And a classical example, like an handbook example, is about the head cell. And so if you imagine a rodent that is moving and is moving also his head, the position of the head is represented by this ring model, and the bump that you could see is moving in the direction of the head that is moving. So it is a sort of a relationship between the bump activation and the head position. This is the meaning of to have the bump as a cell assembly. So to represent a grade, so an angle in the space. And well, this is okay. The main concept. We go directly in more detail. This is the set that we use to represent our model. So basically, we have a simple CA, simple cell assembly, and divided by specific concept. And then also we have cell assembly for temperature. That is the one that we adopt as a physical concept. And we used to have an association, so because there are associative memories. And in particular, we adopt a model that is called triadic, or let's say tree association memory, where basically where you want to activate one concept like hot, you need to activate before other two concepts that themselves activate the first one. So in this case, if you want to activate the hot label, so you need to have activated the coffee ones, and also the degree that is associated with the hot labels. Is a triadic model for to have these associations. And in particular, we have this model where we have the input temperature, that is the physical temperature that is sensed by our sensor, and then we have the specific representation of the temperature for each beverage. So we have the temperature for the Coke and the temperature for the coffee. And then we have this triadic association that is in between the other labels, so the judgments and the beverage. You see that the temperature is different for the Coke, for the coffee, for the Coke. This is because as our nowadays or usual sense of this beverage, we have different kind of tests about the temperature. So in this case, the cold coffee is from 0 to 24, but it's different for a cold Coke that is only from 0 to 10. So there is a sort of difference in this judgment. And in particular, if you look at the bump attractor model that we use is a specific bump, because you can have a different kind of model for the bump attractor. And to represent the temperature, we adopt a 1D dimension of the network. And basically you have taking one neuron, this neuron is able to make positive connection to the closest two ones, you see, and then also the negative, so the inhibition to the 4 beyond. So in this case, the inhibition is a surround inhibition, but it's not totally, but is only limited to the 4 ones after the 2. And we adopt an integrated infer model, and the weight that we adopt is 0.08 nanosiemens, and I can show you more in at the end why. And so this is the structure that we use, and now I present the simulations. And So if you imagine the coffee and you want to activate the specific hot semantics label, you see that what happens is that appears some strange behavior. So if you increase the temperature from 70 until 99, at some point around 10, 11, and so on, it splits. This is a very odd situation. So because at the end, what you have is not a unitary concept. So it's not only to have one hot concept, but you have two hot concepts that represent different temperatures. So it's a sort of doubt that has the mind. And so this is functionally is a fragmented concept. And what we did is to modify the previous network, putting two subnetworks that we call overcame and merge. The merge basically unify the two streams try to unify the two streams, and the overcome at the end close the two streams to have only one streams. And you see now the results after this modification. And it is increasing from 70 to 99. It reached the same threshold, that is a splitting threshold. And after that, you see that there are no two streams, but at some point it starts to go in a convergence manner and then finishes with a mono stream, only one stream. So in this case, there is no ambiguity. So you have your hot concept that is unitary and represent the hot coffee temperature. And there is a problem in this because at the end you have the convergence, but the timing to have the convergence is too huge. So it's not biological at all. And in order to do this, we did another modification on the network. So at the beginning, the weights were statics. So there was no learning basically. So there were only static weights. We introduced a Nebbian learning only for the triadic associations. And what we reach is a concept representation less to 300 milliseconds. It is a common threshold to make decision in the brain. So you see, this is a picture taken from the paper. This is a specific case for the warm temperature, not for the hot. And around 100 milliseconds, the streams converge in only one. So this kind of mind has only one concept represented for the warm temperature. That is fine. And of course, if you want to see more, because this is a very specific example about the hot coffee, but we explored in the paper all the several combinations about the triadic associations. And so if you want to see other kind of activation, you can see also the figure 8 in the paper. So having a recap, so associative memory of binary and bump attractor cell assemblies are able to store discrete-like concepts, for example, coffee, Coke, and cold, and so on. But also continuously value concept, because having a bump attractor, you can store these continuously features. And in our case, we use the temperature as a physical example. And the cell fire persistently. And since that, it is they have the feature to be a memory. So they act as a short-term memory. The first result is about a founding, so that we found that this specific network splits. And so is a very odd behavior. And so it's something that for me and for my other authors so was something very new. And the second result is about the convergence. So if we use other two networks, you can make the convergence of the previous splitting behavior. And the third one is that if you want to have a biological time, it's important to have a Nebbian learning. So it's important to have an association by learning, not by static weights. And so, as I mentioned at the beginning, the paper has been published, and you can download the code and other information with this link. So when you will have the slides, you can do it. And if you are new about simulation and you want to simulate our result, you can see before the Nest environment. It is a simulator, and also Pine. That is a framework that interface the backend, so Nest with your models and your structures. And so this is the end. Thank you. Thank you for the attention.
Thank you very much, Alberto. It was a very interesting talk. Now we have time for questions for both speakers. We already have several questions, and if you want to ask more, please write to us. So I will start with one question for Simone for the first talk. So Kadri is asking, asking that there are suggestions that instead of persistent firing during delay activity, other mechanisms such as short-term synaptic plasticity are instead involved in working or short-term memory, or perhaps both are possible. And the question is, have you considered using this mechanism instead, and would it be possibly change the behavior of the model?
Thanks you, Kadri, for the questions. I have considered this short-term synaptic plasticity 2 days ago in the new model, and yes, I consider yes, of course, and change the model and the structure of the model, of course. Thank you.
Thank you, Simone. We would move, maybe we'll do again more question to Simone and then the questions to Alberto. So we have Carmen that asking 2 questions for Simone. One is, how the learning time increases with the increase of the markers? The number of PF neurons and object neurons must be equal.
Thank you, Carmen. The learning time is the exploration time of the robot. So if I increase the number of markers, there is no increase in the time of learning. The time of learning is not exponential, but quite linear. So the learning time is the exploration time of the robot. The number of persistent foreign neurons are equal to the object neurons number, yes, but the green object is not persistent because the green object is for the exit, and the exit does not not the next marker.
Thank you, Simone. And now I have a question for Alberto. So Pina is asking the examples in the presentation regarding specific concepts like coffee, Coke, etc., but how much the model proposes. The proposed model is able to embed other type of concepts, how much generalizable the model is. I think you talked a little bit about it, but if you have more detailed answer.
Yes, so definitely. So the model is generalizable because in this case we use to have an operative example, that is the coffee and hot. Also is a very framed example. But yes, so if you have more neurons, if you have more cell in the same, more networks, so you can embed other type of associations or other type of concepts. So for example, you could store a supermarket and then try different kind of tea, different kind of coffee, and say every time if they taste different, because now we use the temperature, but we can use also other physical quantities. So it is very generalizable. So the use case is the hot coffee and the other kind of beverage and the attributes about the drinks, but you can extend to whatever you want in theory. So
thank you, Alberto. And another question for you from Matthias is asking, what does the concrete input to the network looks like?
Okay, the input, also, okay, the concrete input, they are spikes. So there is the temperature about the coffee, let's say, and that temperature receive the input temperature, that is the real physical temperature. As you have on your fingers some sensor about the skill temperature, and then it sends on the specific centers in the brain that process the specific temperature of the skin. But the same temperature could be analyzed to another structure in the brain. So we have this input that is only one, that is the physical temperature, and then this input goes directly to several kind of networks. And in one case is the coffee temperature, in the other case is the Coke temperature. But it's only one, the input, that is the physical temperature.
Thank you, Alberto. And we have another question for Simone. Ina is asking this question. She is asking, why is the network able to learn in a single phase sequence of any length?
Thank you, Tina, for the questions. There is two main reasons. The first one is that the SDP rule fitting well in this biological model and works better than backpropagation, for example, or other learning methods of neural network. The second reason is that the network needs also to anticipate the next visual input and not the two next, the three next, and these simplify a lot the model.
Thank you, Simone. And there's another question I see in the chat for Alberto. Julia is asking, she's saying that there are some critical thresholds about the splitting dynamics shown in the video, or do they change randomly?
Okay, so they are fixed. And I have also one slide about this because it's a topic that is behind the paper, because we have also another paper about neuromorphic simulations and so some exploratory simulation that we did about different parameters of the networks. And in this exploration, we found different kind of behavior. So I show you one splitting, so you have two streams, but changing the weights, you can observe several streams or some diverging behavior, like a pyramid that is increasing, or you can have also a splitting pyramid. So it's very odd, and it's very interesting because I think that the theory behind is about dynamical system theory. So where you can have different phases of your systems, and in one case you have one phase with two streams, another phase with three streams, and so on. So it is I think a pretty interesting topic. And in our case, yes, we have two streams, and the threshold of the splitting is about 14 degrees. So if you arrive until 13, there are no problems. If you add on 1 degree, they splits. And it's very constant, static, and so there is no randomness. It's about the structure of the network. So if you change the weights, it changes. But if you don't change the weights or the number of neurons, the behavior is always the same.
Thank you, Alberto. We have one more question, a person that wants to talk. His name is Sudarshan. So we'll give you the opportunity to talk. I don't know if you can hear us. If you can hear us, you can also post your question in the Q&A. I'll continue. There are some more questions. So Carmen is asking for Alberto. Have you studied only triadic associations?
Yes. Yeah, because we assume that to have an activation of one concept, you should have two concepts activated before. Because since it's a judgment, you have an object, an attribute, and a motivation to have that attribute. So you have the temperature, that is the physical quantities, the attribute about the physical quantity that could be hot, and then the object that has that specific attribution, that is the coffee. So there are three elements. Making this triadic association, you can test the network as with this aim.
Thank you, Alberto. And last question for you is by Owen. He's saying that in spiking attractor networks, neurons are often either silent or fire at the maximum rate permitted by the refractory period. Looking at your raster plots that you presented, this appeared to be the case for your model. Did you consider trying to obtain more biological rates?
The model we adopt is a point model. So, and it is a simplified way to build to see how the neuron works. And so, yeah, there are different kind of model, more complex, like compartment models, and also different kind of point model that are able to represent better the functionality of the neurons. So in our case, we used to have spikes that runs every 5 milliseconds. So it's very specific as a period. And what we see is that there is a threshold about the spike in activity. So you can have you have an activation only after 3 degrees. So if you send only one input, the network stay in the resting state. When you reach 3 degrees, it starts, and it starts instantaneously, and is persistent. This is why it's a short-term memory. But at the end, as I mentioned before, you receive the splitting threshold, and it starts to stream. So, but yeah, it's a simplified model of the brain.
Yeah, it makes sense. Okay, so I want to thank again both Alberto and Simone for the very interesting talks. Like I said in the beginning, if you have more questions, you can post in the Slack channel. We'll send you now the link in the chat, or you can also send emails to our speakers. And I would want to also give you some announcement for the end of the session. So the next month in February, we have our HBP student conference. You can still register for the conference. We will also send you now in the chat the link with all the info about the student conference and the registration. I think it's a really great opportunity to hear a lot of talks. We have plenty of times and variety of topics, so it's going to be very interesting. Because we have the conference next month, we won't be having a 10 slides session. So the next 10 slides will be on March. It will be on Thursday, March 4th, at the same time at 2 p.m. We are still looking for speakers. So if you want to speak and participate, please feel free to contact us. It could be any young researcher from the HBP. Doesn't matter if you're in the master's or your PhD or postdoc, and you don't have to present a complete idea or paper, just whatever you feel comfortable to give a short 10-minute talk. So please, if you want to do it, feel free to write us down. We'll be happy. And yeah, that's basically all. So thank you again for the speakers. Thank you everybody for attending, and thank you for the questions. And have a great week. Bye-bye, everyone.
Thank you, Tuzi.
Bye-bye.
Bye,
Thank you.
Bye.